This week we begin a module on fluid dynamics. This work will span 5 weeks. By the time you complete this series of modules, you should have pretty good understanding of the extent to which fluid dynamics undergirds animal form and function. You should know the key differences between solids and fluids and be familiar with the continuum hypothesis. You'll be able to understand the importance of the 3 branches of the study of fluids. There's the theoretical, the experimental, and computational, and understand key concepts requisite to the study of fluid dynamics, such as drag, shear, the difference between laminar and turbulent flows, as well as several important ratios, dimensionless numbers, such as the Reynolds numbers. You will have gained some skills and been able to practice becoming competent in working fluid problems that are relevant to biology. And you will develop and analyze a computational fluid dynamics model. It is important for biologists to understand the basics of fluid dynamics if they want to understand form and function for the majority of animals that have ever lived and that currently live on Earth. This is because most of the environments of Earth are fluid environments, largely either water or air, although many microbes exist in blood or intestines, etc., that are also fluid environments. This image illustrates beautifully the essence of planet Earth, notably that the surface is largely a watery habitat. This is a famous photograph known as the blue marble. It's the first full view of Earth and was taken by Apollo 17 astronauts on their route to the moon in 1972. Today's Earth is also enveloped in a gaseous atmosphere that's composed of about 71% nitrogen, 20.95% oxygen, and 0.04% carbon dioxide. And then there are contributions of lighter gases such as neon and xenon as well as water vapor. Both the watery habitats of the Earth, the oceans, the lakes, the rivers, and the gaseous atmosphere are fluids. The continents and islands, of course, are solids, but they make up only about 29% of the Earth's surface. So most of the habitat available for life is in fact a fluid environment. Earth formed over 4.5 billion years ago, and life first evolved in the oceans, perhaps as early as 4.1 billion years ago. The single-celled organisms that first evolved soon began transforming the atmosphere of Earth, for example, by making oxygen. And organisms continue to shape Earth's atmosphere today, notably by the addition of CO2 from combustion of fossil fuels. The metazoa, or animal life, also evolved in the oceans. The oldest fossil record of the metazoa consists of creatures that resemble segmented worms. These date back to the Ediacaran, about 560 million years ago. Animals continue to evolve in an aquatic habitat, and the vast majority have never left this environment. We'll come back and talk about this again in a minute. I've divided that blue marble up into a pie chart that illustrates the number of species that are currently known to exist on the Earth, and the larger the piece of the pie, the greater the number of species in that particular lineage. So this is of all chordates, and of course vertebrates are chordates, but chordates also include the immediate outgroups of vertebrates, so these are the tunicates and the lancelets. So on the inner circle, I've written the lineage, such as fish, and in parentheses it's the percent of species that fish comprise. And then on the outer circle, there's several different types of fish here listed, major lineages of fish, and in parentheses in this outer circle you can find the actual number of species.
So you can see this huge, almost 50% of the pie is composed of indeed fish. And of those, there's some hagfish. There's few, like 80 species of hagfish and lampreys. There's about 900 species of chondrichthyes, so these are the sharks and the rays. And the rest, almost all the rest, are ray-finned fishes. There's over 25,000 species of ray-finned fishes. And then there's another type of fish, the lobe-finned fishes, from which terrestrial animals or vertebrates arise. And there are a few remaining lobe-finned fishes that include the coelacanths and the lungfishes. So there's only 8 species of those. Amphibians, you can see, make up about 9% of chordate diversity. Birds make up about 17. There's around over 9,000 species of birds. 9 avian reptiles are 13%, and mammals make up about 8%. Here I have covered the groups that are moving through a fluid in blue. So you can see the huge percent of this pie that consists of animals that are either flying or they're swimming. So all of the tunicates are aquatic. The majority of fish on this planet are aquatic. There's a few that come out of the water part of the time, mud skippers and things. The majority of amphibians are aquatic at some point in their lives. There's a few toads and others that are completely terrestrial, but they're a minority. Almost all birds fly. You can see that of the mammals, bats are the most speciose, and of course they're flying. And the non-avian reptiles, most of them, they are the most terrestrial group. Most of them are terrestrial, but even so, I don't know, 8 or 9% are probably semi-aquatic or aquatic. So all of the crocs, for example, are aquatic, and some turtles, marine turtles, are completely aquatic and things. So that is the blue portion of this pie is actually a little bit of an underrepresentation, because I didn't try to figure out exactly how many of these non-avian reptiles are aquatic or semi-aquatic. And you have to remember, some of the mammals that are marked as being terrestrial, those that are included in that wedge of the pie, are actually aquatic. So we have a lot of cetaceans, 89 species, 33 species of pinnipeds, very few number of rodents are aquatic, etc. And then there's some rodents, or some mammals that also glide. So more than 60 species of mammals are moving through the air. So you can see that understanding how animals locomote through a fluid is really important to understanding why they're built the way they are. While more than 82% of chordates are moving through a fluid, 96% are convecting fluid in a respiratory system. So they're doing gas exchange by either moving fluid over gills or pulling it into and pushing it out of lungs, etc. So understanding how fluid interacts with structures internally, how it flows through tubes and whatnot, is very important in terms of understanding the design of the respiratory system. 100% of chordates convect fluid in a circulatory system. So even the handful of chordates that didn't convect fluid for respiration, so those would be animals like the giant salamander that just does respiration over the skin. They've completely lost the Japanese salamander, they've lost their lungs. Or tunicates and lancelets, they rely on the skin again for respiration. All of those animals still have a circulatory system, so it's 100% of chordates that need to be able to push fluid through tubes in a circulatory system. I would like you to watch a couple of videos of locomotion on land compared to locomotion in water. So this first one is of a greyhound running on a beach. And the reason I want you to watch these is because I think they really illustrate beautifully some of the key differences between these two different types of modes of locomotion. Now recall that we talked about the fact that when you apply a force on a solid, it's important to think about how that force is distributed over an area. So how large is the surface area of that foot or those feet as they touch the earth? And force divided by area, of course, is stress. So we really want to think about the stresses on that musculoskeletal system. What I'd like you to really take home here is that these stresses are being applied throughout different phases of this locomotor cycle to a single limb. So they're tremendous. Because of that, dog putting all its weight on an individual limb one at a time like this, it's loading those limbs with a tremendous amount of stress. Recall we talked about 3 different types of stress. We have the linear stresses of compression and tension, and then we have, and they result in linear strains, so the bones and muscles are compressed together or pulled apart. And then we have the shear stress. And the strain that it produces is angular. It's changing the angles of the structures within the solids, within the bones and muscles. And it is this latter stress, the shear stress, which is really, really different in water compared to, or air compared to land. So let's have a look at that. The next 2 videos show locomotion in water at high Reynolds number and at low Reynolds number. We're going to come back and talk about Reynolds number in a minute. And but for now, I just want you to know that these are 2 distinct types of locomotion, largely because of the Reynolds number. And I've also put a link on this slide to a 3rd video, which I'm not going to play, but I recommend that you watch this. It's called Human Sperm versus the Sperm Whale. I also have a link on Canvas. And this does a really excellent job at explaining Reynolds numbers, what the number is, the history of it, and why it's so important in understanding movement in fluids. So I'm not going to talk during this first movie. I'm going to play the one by the BBC because it's narrated. But what I'd like you to notice is the way that these different species move in water at high Reynolds number. Okay, so you're going to see several species of fish, and they use what's called lateral undulatory locomotion. And you're going to see 3 different species of marine mammals, and you're going to see a bird, which literally flies underwater. So think about the stresses that the musculoskeletal system is experiencing.
Even when more sea lions arrive, they can't seem to break down the sardines' coordinated defenses. With a shoal this big, the sea lions need to isolate a smaller, more manageable group of fish. But with so few predators, the fish still have the advantage. All the sea lions can do is keep the sardines at the surface. and wait for others to join them. Tuna. Their arrival changes everything. Tuna attack from below, cutting off the sardines' escape route down to deeper water. Next to appear shearwaters. Excellent flyers, but also surprisingly agile underwater. With so many predators attacking from all sides, the advantage starts to shift away from the sardines. As the fish pack ever tighter, their shoaling strategy now makes it easier for the hunters. Copper sharks. They've scented blood in the water. Surprisingly, perhaps, the predators never attack one another. They work together to corral the ball of fish, taking turns to grab a mouthful. Common dolphins. As the shoal gets ever smaller, each sardine scrambles desperately to hide in the middle. But now there's no escape. A brooder's whale finishes off the feast. Tons of sardines devoured in less than an hour.
Now observe organisms that are using flagellar locomotion. And the thing that is noteworthy here that I'd like you to pay attention to is that some points in time, they actually look like a corkscrew that you would use to open a bottle of wine, right? The first step to understanding the differences in these locomotor modes is to either review for you or to introduce you to some of the basic physics of fluids. By far, the most important property of a fluid is that it is unable to resist deformation under an applied shear stress, no matter how small. Furthermore, fluids don't resist permanent deformation. They resist the relative rates of deformation, and they do this in a dissipative or frictional manner. These first two are very important. Fluids also can flow or take the shape of a container. They respond to normal stresses, compression and tension. And for gases, they have no free surface, so they're going to freely diffuse out of their container if the container is open. Whereas liquids and solids have a free surface, and this surface minimizes the energy state. So for a solid, it is a crystalline structure, and for liquids, it's surface tension or droplets. Some substances have part of the properties of fluids, but they're not actually fluids. A good example is sugar. You can pour it from a cup, it flows, it takes the shape of its container. However, because you can pour it into a pile, that means that it's resisting small shear stresses. Whereas coffee, you pour that onto the table and it's going to form a puddle, right? There's not going to be a pile of coffee. So, again, the key here is that a fluid is a substance that deforms continuously when subjected to a shear stress, no matter how small that shear stress is. This slide illustrates beautifully an important property of fluids, and that is their ability to form a vortex. So the great red spot of Jupiter is a vortex, and you can see some of the swirling bands in the bands of Jupiter. Those are vortices. And also you can see that in the clouds. These are photographs that are to scale, by the way. I think they're stunningly beautiful. On the bottom of the slide, I put a quote by one of my heroes, scientific heroes, a physicist named Richard Feynman, who actually began his career studying fluids and then gave up on it. It was like, that's just too complicated and too difficult. So he moved into particle physics and did some work that ended up winning him a Nobel Prize. He's known for a book that he wrote that is popular. It's called Surely You're Joking, Mr. Feynman. And it's about his life, but a lot of it has to do with his work on the Manhattan Project. He helped out with developing the atomic bomb during World War II. But anyway, he says, everything that living things do can be understood in terms of the jiggling and wiggling of atoms. And although technically true, it's pretty difficult to keep track of the velocity, the direction, the momentum of every single atom in a solid, but even more so in a liquid or a gas. So technically this is true, but there's a better way to do it, and this better way is known as the continuum hypothesis. What the continuum hypothesis says is that, in sort of simple terms, it says that when dealing with fluids, we can ignore the fact that they actually consist of billions of individual molecules in a rather small region, and instead treat the properties of that region as though it were a continuum. So each one of these regions is known as a fluid parcel. It's also called a fluid element or a fluid particle. So it's a region or a point in a fluid that has its own set of properties. It could be temperature, it could be velocity, it could be pressure, it could be momentum, and the fluid as a whole then is seen as an aggregation of each one of these fluid elements. So one of the first questions you have to address, and you'll address this a little bit in your fluid dynamic work, is how big should a fluid element be? If you have a fluid in a certain volume, how many little volumes do you want to divide that fluid into? And the answer has to do with your measurement in each one of these fluid volumes being statistically significant. So if you only have one molecule in that fluid volume and you're trying to measure temperature, the molecule might not even hit your temperature probe. So that's clearly not enough molecules in that fluid volume. The volume needs to be bigger. And then the other thing that we're going to look at is, and you have to consider, is how do you measure or compute stresses and strains on each one of these fluid particles. So let's take this first question. How big should a fluid element be? And I think that it becomes really clear when you have a concrete example. So let's say you wanted to know, you want to measure something about air, or calculate it, estimate it. Air is made up of mainly nitrogen and oxygen. Both of these are ideal gases. And so let's just consider air being composed only of those to make it simple at standard temperature and pressure. How many molecules are in, let's say, if you made your fluid element a cubic meter, so each length of that cube is a cubic meter. Well, you need to know the density of air and the volume that a mole of air occupies at this standard temperature and pressure. So the volume of that cubic meter contains, it turns out, 1,000 liters of air. And each liter, there are 22.4 liters in a mole for an ideal gas. So 1 mole is going to take up space of 22.4 liters. And there's Avogadro's number in 1 mole, so that's 6 times 10 to the 23 molecules in that mole, which gives you, in that cubic meter, 10 to the 25 molecules. Okay, so that's a ton. That's more than enough to be statistically significant for whatever you're measuring or computing. So how about if we go down to a volume, a fluid element that is a centimeter on each side, length of the cube. You do the same thing. You can do the math. You know how much volume is taken up by 1 mole, how many molecules in a mole, and what you see is in that cubic centimeter, and a cubic centimeter is a mil, right? That's 10 to the 19 molecules. So in 1 mil, or 1 cubic centimeter fluid element, there's more than enough molecules to be statistically significant. Go down another order of magnitude to a micrometer. Do the same math, and what you find is there's still plenty. It's 10 to the 7 molecules, so it's huge. Go down to a nanometer scale. So each side is a nanometer. And what you find is in your fluid element, you're going to have 0.02 molecules. So that's clearly not going to work. At nanometer scales, then, the continuum theorem or hypothesis is no longer a valid approach. And so if you wanted to study the flow of water or solutes through an ion channel, for example, well, a membrane is around 4 nanometers in thickness. And water is a lot more dense than air. I think it's a little more than 800 times as dense as air. But even so, a cubic nanometer of water is going to have 0.02 times 800. So that's still not enough molecules. I don't know what that is, 22 or something. That's still not enough molecules to be statistically significant. So at these smaller scales, at the nanometer scales, you have to switch to a different method, a different theory, and that's diffusion. You have to start worrying about each and every one of the molecules or atoms in your system. So you can't just lump them together into fluid elements anymore. But you have to keep track of each and every one and deal with them using the theories of diffusion. The study of forces acting on a fluid and the ability to estimate fluid behavior requires an understanding of 2 classes of properties of fluids. The first are transport properties, and these include viscosity, thermal conductivity, and mass diffusivity. Viscosity is the one we're going to focus on. It's the most important. So it's the one we're going to focus on. And again, I hope you've already watched this video on Reynolds numbers and viscosity. The other class of properties of fluids are things like density and pressure. So viscosity is like an internal friction. Highly viscous fluids are sticky or gooey, for example, honey, molasses, tar. Those are all highly viscous fluids. So you have fluid parcels or elements within that fluid, but they don't want to move, slide past each other readily. They're sticky. They resist that with a high internal friction. They resist that flow. So if you take a container of molasses and try to pour it out, it's going to flow very slowly compared to that same container, the same opening, poured in the same manner if the fluid in there is water, for example. So viscosity is that fluid property by virtue of which a fluid offers resistance to shear stresses. That's its formal definition. There are many fluids in which the rate of angular deformation within that fluid is directly proportional to the shear stress that is applied and the viscosity. So Newton's law of viscosity, which doesn't apply to all fluids, but says for a given rate of angular deformation of the fluid, shear stress is linearly related, directly proportional to viscosity. So fluids that are Newtonian are things like water and air. Non-Newtonian fluids include blood and solutions of cornstarch. We'll come back to these. Just as we learned that when you're applying a force to a solid, the area over which that force is distributed is quite important. And that relationship of force per area is the stress. And we learned about three simple stresses: compression, tension, and shear. I want to start out with these first two and think about applying these stresses to fluids. So there's lots of biologically relevant examples of fluids loaded in compression. You can think of the heart contracting and compressing blood that's contained within the heart. There are changes in compressive forces as animals move to different altitudes. So we are constantly under compression from the atmosphere. The molecules of the atmosphere are pushing down on us. But if you go to high altitude, and there are birds, condors fly around at 30,000 feet. This is the same altitude that airliners fly at. So they're under greatly reduced compressive stress when they fly at that altitude. It makes a big difference in terms of the lift that they can generate, in terms of the way that air flows throughout their respiratory system, etc. And then for aquatic animals, they're under greatly increased compressive stresses because they not only have the weight of the atmosphere pushing on them, but they also have the weight of the water column that's above them, all the mass of the water molecules. And you can compute that pressure, that compressive stress, pretty readily. I gave an example of how you would do that. So let's say you wanted to compute the pressure in the center of this fluid-filled container. What you do is, first of all, you have the baseline of the atmospheric pressure, P naught, and then you have the pressure of that water column, which is given by the product of the density times the height and the acceleration due to the pull of the Earth's gravitational field. So that's 9.8 meters per second squared. So again, a pressure is a force per area. So you're going to get, when you make these calculations, you're going to get something like force in newtons, right, divided by an area. So it would be meter squared. So some biologically relevant examples of tension fluids include plant transpiration and the flow of water through these tubular structures in plants, the xylem, and also the example of cavitation. So plants have these little openings on their leaves, these stoma or stomata, plural, and they open and then water evaporates, and then these the adjacent water molecules are pulled towards the opening through capillary action, and this can set up a negative pressure within these rigid structures, and that will then suck up, literally suck up water from the roots. Because the water is under reduced pressure, any gases that are dissolved in that water can start to come out of it and form bubbles. And if there's big enough bubbles that coalesce and get together, it can actually cause what's called cavitation, and so it stops the flow of this water. This provides another interesting example of where hydrostatic pressure, or this compressive stress, is important. So let's say you want to know what is the stress, what is the pressure the heart needs to generate in order to pump blood up to the height of the brain of a giraffe or a sauropod. You can use the same equation to compute what that stress needs to be. So it's the density of the blood, the acceleration of gravity, and the height that the fluid has to be pumped up. I'm going to end the first part of this lecture here. It's to be continued. We're going to move on and talk about this third simple stress, applying a shear stress to a fluid. It's incredibly important. But I'm ending this lecture here because I'm embedding so many movies that I'm having problems with the movie starting to play, etc., etc. So I'm not going to break this PowerPoint into two different lectures, but we will pick this up in part 2.